Assalamu alaikum. So let's start with the molecular biology course. Or the molecular biology part. I'll go over what we covered in biochemistry, and then I'll talk about a few techniques. Well, I'll start with a basic technique called gel electrophoresis. So let's start. So DNA is a polymer. It's made of four monomers um, that are different from each other by the bases that they contain, that they're made of. So you have cytosine, designated as C, guanine, designated as G, thymine, T, adenine, A. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. The nucleotide is made of, in addition to the base, each nucleotide is also made of a sugar, a ribose sugar, a deoxyribose sugar, and a phosphate. DNA is a double helical molecule. So you have two, what we say, called strand, and each strand is made of these monomers, these subunits, that are linked to each other by a phosphodiester bond. Now, so each strand is made of this sequence, okay, not the word that I'm using, a sequence of nucleotides. So it's a double helical molecule. Helical because you have the two strands going around each other, they wind around each other in a helical pattern. But it's not really a perfect helix. It's not a perfect helix. So you have these rooms right here. You have these spaces, and these are called grooves. So if you look at the DNA molecule, you see that there is a major groove followed by a minor groove, then a major groove, then a minor groove. Opposite to a major groove, you have a minor groove, and opposite to a minor groove, you have a major groove. And these groovings are important because that's where proteins fit when they interact with DNA. So you have these two strands, and these two strands are anti-parallel. But before we talk about the anti-parallel feature of DNA, let's talk about the base pairing. So you have two strands opposite to each other, sort of, and you have this base pairing. So C pairs with a G and A pairs with a T. Now the pairing between C and G is mediated by three hydrogen bonds. The A and T pairing is mediated by two hydrogen bonds. And this is important because if you have DNA that has a lot of G's and C's, this means that the two strands are connected to each other in a stronger manner than a DNA molecule that is rich with A and T. Now, so because of this very specific base pairing, note I'm using the word base pairing. So here you have a base pair. So each base pair each base pair consists of two bases or two nucleotides that are paired to each other by hydrogen bonds, and we say that they are complementary. So they complement each other, become لبعض. So they complement each other. So A complements T and C complements G. Note that the base pairing is always between a pyrimidine base or nucleotide and a purine nucleotide. So here you have purine and here you have a pyrimidine. In each strand, there is a backbone, and the backbone is the phosphate followed by the sugar, then phosphate, then sugar, then phosphate, then sugar. And you have the side chains, and the side chains are the bases themselves. So they go sort of perpendicular, amudi, to the backbone itself. Now the two strands are anti-parallel to each other, because note, what do we mean by that? So the phosphate group, this one right here, is connected to carbon number 5 of the sugar. So we say that this is 5 prime end. And at the end of just one strand, you see that the base, the sugar has a free carbon number 3, which we call the 3 prime carbon. So there is a 3 prime end. So here you have a 5 prime end on each strand, and you have a 3 prime end. Now, if you look at the opposite, the complementary strand, you note that here you have the 5 prime end, and here you have the 3 prime end. Okay. Now, what else? DNA is a stable molecule, yet it's flexible because it can bend, but it's really hard to break up the phosphodiester bond. Now, this is how we write the sequence of a DNA molecule. Here you have the 5 prime end, and here you have on the same strand the 3 prime end. Now, the complementary strand again is anti-parallel. So here you have the 5 prime end, and here you have the 3 prime end. And the bases are complementary to each other. So A pairs with T, T with A, G with C, and C with G, and so on. Now, whenever we can write the DNA like this, by the way, without indicating the 5 prime end and the 3 prime end. So if I say that this DNA sequence is A, T, G, G, C, C, we should all know that we're starting the reading of the DNA sequence at the 5 prime end. So this is given, this is a rule. Now, I don't really have to write, if I say DNA, we should all know again that it's double-stranded. And I do not have to write the complementary strand, the anti-parallel strand. So I should know that this is the 5 prime end, and here we have the 3 prime end. And we should know, it's given, that the complementary strand starts with the, well, it doesn't start, but we write it 3 prime end right here, 5 prime end right there. And I know, you should all know that complementary to A is a T, complementary to a T is an A, complementary to a G is a C, and so on. Now, on the other hand, by the way, we'll talk about RNA later on. RNA has A, G, and C in addition to a uracil. So instead of a T, there is a uracil in RNA, and usually RNA molecules are single-stranded. They're not double-stranded like DNA. Now, if I say genome, I'm talking about the whole total genetic material of a living being, or like bacteria, human, a species like monkey genome, human genome, or an individual. So it's my genome, your genome, and so on. Or we can say that's the genome of the brain cell or the liver cell. Now, if they are from the same individual, they should be identical, theoretically speaking. The prokaryotic bacterial genome is organized as a single circular molecule of double-stranded DNA. In addition to the chromosome, the single chromosome, Bacteria also have smaller genetic material known as plasmid. So they are circular double-stranded DNA as well. And usually bacterial cells have one genome, one chromosome, but they can have multiple copies of the same plasmid. Eukaryotic genomes, on the other hand, including the human genome, is organized as the human genome is organized as linear chromosomes. And these chromosomes are made of, again, long double-stranded DNA molecule. Each one of them is made of single DNA, double-stranded DNA molecule. And there are histones that pack the DNA. Eukaryotic cells also have mitochondria, and mitochondria have their own genome as well. And the mitochondrial genome is less stable than the nuclear genome. So these chromosomes exist inside they are enclosed by a nuclear membrane. Now the mitochondria, okay, somewhere in the cytosol there are multiple mitochondria, and each one of them can contain multiple genomes, and these genomes are sort of identical to each other. So this is what we will be talking about. We'll talk about the organization of the chromosome, of the genome. We'll talk about DNA replication process. We'll talk about transcription. We'll talk about the different types of RNA molecules and what they do. We'll talk about the process of translation. Okay. At the end of this part of molecular biology, we'll talk about mutations and repair. Throughout our lectures, we'll talk about the major techniques that we can use in molecular biology. So, and these techniques will be scattered along the lectures. So whatever the technique fits, I'll talk about it, and we will build up information based on that. At the end of the molecular biology part, we'll talk about it during this lecture of DNA repair. We'll talk about the exciting technology of CRISPR-Cas9 and the potential it holds in medicine and in science overall. So I hope you'll enjoy the journey and the molecular biology part. Now, the processes that we will talk about: DNA replication, transcription, translation. That is, DNA makes RNA by a process known as DNA makes DNA, okay, by a process known as replication. DNA can also make RNA by a process known as transcription. And then RNA can be used to make proteins by a process known as translation. This is known as the central dogma of molecular biology, العقيدة الأساسية للبيولوجيا الجزيئية. That is, DNA makes DNA, DNA makes RNA, RNA makes protein. But we will learn that there are exceptions, that RNA can make RNA, RNA can also make DNA. So we will briefly talk about these other processes as well. But I want to remind you of something that is really important, and that is proteins can interact with DNA. So remember that we have these bases, and these bases are sort of hidden between the helical strands of DNA, except that some groups of the bases can be exposed outside, and they can be seen by protein molecules, specifically the amino acids that make up the proteins. And here we're talking about the R groups of the amino acids. Like, for example, the carboxylic group of glutamate, the carboxylic, the hydroxyl group of serine, the amino group of asparagine, okay, and the amino group of the lysine as well, the positively charged amino acid. Now, these are the groups of amino acids can interact with the bases of DNA. And these interactions can be really specific. The order of the bases, as well as the overall three-dimensional structure of the amino acids, can determine the specificity of interaction between proteins and DNA. So overall, proteins do not randomly interact with DNA. Rather, they recognize specific DNA sequences, that is, the order of nucleotides within a DNA strand, a DNA molecule, and the interaction between these bases and the amino acids are quite specific. So remember that when we talk about DNA protein interaction. Probably an exception to the specific DNA protein interaction is the interaction between histone molecules and DNA. So our DNA, by the way, in each individual cell is really long. It's about 2 meters long. And this 2-meter strand must fit into this really small, tiny nucleus. And this happens because the DNA molecule is wrapped around the histone molecules, the histone octamer that we talked about in biochemistry. So the DNA is wrapped around it, okay? And it looks like beads on beads that it looks like a string that wraps around beads مثل الخرزات والخيط زي المسبحة. So that's how it looks like, and the DNA can be really packed. Now the interaction between DNA molecule, the DNA molecule and histones is known as chromatin. The molecule is known as a chromatin molecule. And we talked about nucleosomes and the different structures of that exist in DNA molecule. All right. So again, there are differences between DNA and RNA, and these differences include the following. Overall, DNA is double-stranded, RNA is single-stranded. DNA contains a deoxyribose sugar. RNA contains a ribose sugar, and that's why DNA is known as deoxyribonucleotides, and RNA is known as ribonucleotides. And here we're talking about this group right here. This is the one that is deoxygenated. It's the one that is linked to carbon number 2, the 2' carbon. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. The phosphodiester bond before. So here you have a hydroxyl group. Okay, so it's not deoxygenated. So it's a ribose molecule. This is a deoxyribose. The phosphodiester bond takes place at this hydroxyl group that is connected to carbon number 3, that is the 3' carbon. And here you have the 5' carbon right here. Now, the bases that make up DNA include thymine, cytosine, adenine, guanine. Thymine does not exist in RNA, rather we have uracil. In addition to the 3 common ones with DNA, cytosine, adenine, and guanine. Now, whereas our cells contain single DNA molecule that is organized as chromosomes. We have different types of RNA molecules. And in fact, we have many, many types of RNA molecules. Of course, we know the three major ones. The messenger RNA that is used to produce proteins, transfer RNA that is also used to produce proteins, except that tRNA molecules are small RNA molecules that have amino acids linked to them. And we have the ribosomal RNA, which are important in the structure of ribosomes, which are the factories of protein synthesis. And the ribosomal RNA molecules are responsible for forming the peptide bonds between amino acids to make proteins. Now we have different types of RNA molecules. But in these lectures, I'm just going to talk about two of these RNA molecules, and they are microRNA, microRNA, which are important for regulating protein synthesis. And we have long non-coding RNA or link RNA, which have RNA molecules and they have different functions, such as they regulate transcription, they can regulate the processing of messenger RNA, they can regulate the abundance of microRNA, and they can control microRNA function, and they are important in determining protein function. So we'll talk about long non-coding RNA as well during these lectures. So this is an overview of what we sort of covered in biochemistry and what we will be talking about in this part of the course. So let me start by introducing a few techniques. Probably in this recording, I'm going to cover one of those techniques. But first, I want to differentiate between two terms. And these terms are DNA staining and DNA labeling. DNA staining and DNA labeling. So DNA staining is basically if we take a shirt and we stain it with paint, for example. So we give a color to the shirt. Now this color, we can see it with our own eyes. And this can happen with DNA as well. So we have this DNA molecule, and we can add certain chemicals, and these chemicals can be inserted between the bases of DNA, and they can give us a certain color. Now, DNA labeling, on the other hand, is a bit more sensitive, by the way. And basically, whereas here in DNA staining, what gives a color is the molecule that is added to the DNA, and this molecule is separate from the DNA molecule. Here we have the DNA itself giving or emitting a signal, a fluorescent signal, and the signal can also be radioactive. And this signal is quite sensitive, by the way. Now, so here we have the DNA itself emitting, releasing the signal. So the signal, for example, we can use radioactive phosphorus, and radioactive phosphorus can be inserted in the DNA when DNA is synthesized. So, so the DNA itself gives out the signal, or we can attach covalently a certain dye, a certain molecule that when it's hit by light at a certain wavelength, energy is released in the form of light at a different wavelength, a different color. So we can hit DNA with a green light, and a yellow light would be generated, and this can be detected. Now, so either the molecule is inserted into DNA non-covalently and it gives a color, or so we stain it, or we label the DNA. The DNA itself gives a color. So it's like wearing this hat that has a light attached to it. So the person itself is not really emitting the signal. It's actually what we add to that person covalently again that gives a signal. So pay attention to these two different terms, DNA staining and DNA labeling. So let's talk about this technique now. Okay. It's called gel electrophoresis. And the idea here is that we form a gel with a certain three-dimensional structure. And it looks like this. It's like, again, it's like jello. Okay, so it's liquid and then it solidifies. And when it gets molded in a specific shape. And this shape looks something like this, flat, like the palm of your hand. Okay, and there is like a hole, not exactly a hole, but we call it a well. So we can add a sample inside this well. Okay, this is a top view of the gel, and here we have different wells. So we can add sample 1 here, sample 2, sample 3, sample 4, sample 5, etc. And the gel looks something like this. So you have a network of chemicals, and there are spaces in between. So when we add DNA to a well and we apply an electrical current, here we have the cathode, the negative side. Here we have the anode, the positive side. Now DNA is negatively charged. What does make it negatively charged, by the way? Find out what the answer is. So we add DNA and we apply an electrical current. Now, since DNA is negatively charged, it's going to run away from the negative end. So it's going to move towards the anode. The smaller DNA molecules would travel faster than the larger DNA molecules. And here we're talking about the length of the DNA, the length of the strand of the DNA. So the smaller molecules are going to go through these holes, these pores within the gel faster than the larger DNA fragments. So let's say here we have DNA, we apply a current, negative, positive, and the DNA molecules are going to travel inside the gel. Okay, so the larger molecules are going to travel or migrate or move or run, as we call it. They're going to run slower than the smaller molecules. Here's another view right here. So here we have the DNA molecules inside a well. We apply electrical current. Again, the smaller molecules are going to migrate, run, travel faster than the larger molecules. Now, so again, here we have the wells, and we have sample 1, sample 2. Now, in this well, we add what we call a size standard, which is basically a sample that we usually purchase or we prepare in the laboratory. And it contains DNA molecules of different sizes. And we know what the sizes are. So we know that this molecule, for example, or this band, so each the DNA molecules are going to travel as bands when stained. So here we have the DNA run, we stop the electrical current, we take the gel and we stain the DNA, we color it, and we look at the color. Now each band contains many thousands, billions of molecules of DNA. They have the same size. They can be of different types, but they have the same size. So each band does not contain one DNA molecule, rather each band contains, again, thousands to millions of DNA molecules. Now, we take the DNA and we stain it. And here we have a standard. We know that the size of the DNA molecules inside this band are of this size. Note how we call it: 1,000 base pair, meaning that each strand contains 1,000 nucleotides. The complementary strand also contains 1,000 nucleotides, so together 1,000 base pairs. This band, we know that it contains DNA molecules of this size: 850 base pairs, 750, 600, and so on. So note how they travel. The smaller DNA molecules travel faster than the larger DNA molecules. Now, we have these two samples, sample 1, sample 2. We add the sample here, we apply electrical current, the DNA molecules are going to travel toward the anode. And when we take a picture of the gel when the DNA is stained, this is how we look at it. Okay, we look at it from the wells should be on top, and the molecules sort of travel downward, but rather, when we do the technique, the DNA travel horizontally, ufuqi. Okay, so I know that these DNA molecules within this band travel at the same distance as this band right here. So I can estimate that this DNA molecules within this band are about 1,000 base pair. Okay. We know that the same thing applies here. Okay. Now we know that this one here is about 850, and so on. We know that this one here is probably 750. This one here is between 750 and 600. So it's probably closer to 700 than 600, let's say, or 650. It's just an estimation of what the size of the DNA molecules inside this band is. Now we can estimate the size of this DNA molecule. We say, aha, it is smaller than 600, and it's a bit larger than 200. So I would say it's about 500, maybe 450, and so on. So we can estimate what the size of this band is. Okay, so this is gel electrophoresis, and that's how we can analyze DNA. So keep that in mind, because we're going to talk about, I'm going to show you a lot of images later on of gels, sometimes illustrations. So keep in mind how we look at these gels and how we estimate the sizes of the bands and what these bands really mean. Now, if the amount of DNA in a sample is very little and we cannot really see it when we stain it, we can label the DNA. We can use radioactive phosphorus, for example, and it gives us a much stronger signal, okay, allowing us to provide us. It can provide us with a more sensitive technique, a more sensitive approach to detect DNA. Now, there are many YouTube videos and whatever websites that you can search for to see how it looks like, how gel electrophoresis is, and how it works. I'll try to show you a gel, inshallah, in the next lecture when we meet together, so that you can comprehend what a gel looks like. Okay, and inshallah as well, I'm gonna download or upload. I'm gonna upload a few animation videos of gel electrophoresis, so you can watch them as well. So how do we measure how much DNA we have in a sample? Now, remember that DNA, that we cannot see DNA. In reality, we cannot really see DNA because it absorbs light in the UV range, and we cannot see if something, we cannot see something that absorbs light in the UV range. The reason why my shirt is blue is because it absorbs the blue color and it reflects everything else to your eyes, and that's why you see the blue color. So what DNA does is that it absorbs the UV light. Now, we cannot really see the UV light, and as a result, we cannot see DNA, and that's why we have to stain it. That's why we have to label it. Okay? Now, in order to measure the amount of DNA in a sample, we use an instrument known as a spectrophotometer, and the technique is known as spectrophotometry. And the principle behind this instrument is that we take that DNA sample, we put it in a vial, and we hit it with light. Specifically, we hit it with a UV light. So all wavelength of light would be distracted except for the UV light that hits the DNA. The DNA absorbs the UV light. And then the amount of absorption can be detected by a detector or a source. So we have a certain figure that the instrument gives us, and the instrument tells us that this amount of light is absorbed by this sample. And then we can convert the amount of light that is absorbed to a concentration. So we know the following. We know that if I have 1 ml of sample and this sample contains 50 micrograms of DNA, so the concentration of the sample is 50 micrograms per ml, we know that this sample is going to absorb 1 unit of light. So again, 1 unit of UV light would be absorbed by DNA that has a concentration of 50 micrograms per ml. So the question is, what if the DNA sample contains a concentration of 25 micrograms per ml? What do you think the amount of light that would be absorbed? It's half a unit. Of course, half the concentration of DNA would absorb half a unit of light. Okay, now think about this. What if the amount of light that is absorbed is 0.1? That is 1/10. What would the concentration of the DNA sample be? Think about it. So, sometimes I have samples that contain DNA and really large amount of DNA. So the DNA is really concentrated. So it's about, let's say, 500 micrograms per mil. That's a lot of DNA. Now the instrument, the spectrophotometer, cannot accurately calculate how much light would be absorbed. There's a limit to this instrument that it can measure. So what we do is that we dilute the sample. بنخففه. We use a diluted sample. How do we do that? Well, I have a sample that has a concentration of DNA of about 500 micrograms per mil. I take 1 microliter. I put it in a water sample that contains 9 microliters of water. So I add this 1 microliter to 9 microliters, that's 10 microliters. So I have 1/10. So I've made a dilution of 1 to 10. So the sample is 1 to 10. The concentration of the sample is going to be, the diluted sample that is, it's going to be 50 micrograms per ml, because now the original sample is 500 micrograms per mil. But if I dilute it 1 to 10th, the concentration of the diluted sample is going to be 50 micrograms per mil. Got it? Inshallah. Now, so we have to take this into account. We have to take the dilution factor into consideration. We have to put it in the formula that allows us to calculate the concentration of the sample, of the DNA sample. So we have this problem right here. What is the concentration of a double-stranded DNA sample diluted at 1 to 10? Aha, so that's the dilution factor. And absorbance, A, absorbance at 260 nanometers, that's the UV range. This is the maximal wavelength that DNA can absorb, is 0.1. So 0.1 unit of light is absorbed by this sample, and this sample is diluted 1 to 10th. So how do we calculate the concentration of the original sample? So we do the following. We know that 1 unit of light means that the concentration of the sample is 50 micrograms per mil. Now the amount of absorbance is about 0.1 unit of light, so it's 1/10 of this. So this means that the concentration is 1/10 of 50. So basically, the concentration of the diluted sample is about 5 micrograms per mil. Well, we need to calculate the concentration of the original sample, not the diluted one. So what do? We multiply by the dilution factor, that is 10. The dilution is 1 to 10. So we say 0.1, the amount of light that is absorbed, times 10, the dilution factor, times 50 micrograms per mil, because this means 50 micrograms per mil over 1, which is this one right here. Okay? That's constant, by the way. Okay? It's constant. We know that 50 micrograms per mil of DNA would absorb 1 unit of light. So, if I have my sample absorbing 0.1, it means that the concentration is 1/10 as well. So, but we have to multiply by the dilution factor, which is 10. Okay, so the concentration of the original sample is 50 micrograms per mil. Okay, so I'm going to stop here. You can practice, just play with the numbers. Okay, change this problem right here into different forms. So you can use different dilution factors. You can use different absorbance units. You can start with a DNA sample of a known concentration and try to calculate the amount of light that would be absorbed or the dilution factor and so on. So manipulate this problem in different ways. So hoping that, inshallah, you will understand it. I'm going to stop here. Thank you so much. We'll continue in person by the end of the week. Allah ma'akum.